Hi, everyone, and welcome to today's webinar of Proteomics Signature of NASH. I'm Dr. Dion Denmark, the Senior Director of Translational Medicine and Clinical Development at SomaLogic, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by SomaLogic. To learn more about SomaLogic, visit somalogic.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here, and if you have trouble seeing or hearing the presentation, please do let us know. So I'd like to now welcome our speaker, Dr. Arun Sanyal, Director of the Stravitz-Sanyal Institute for Liver Disease and Metabolic Health. He is also the Interim Chair and Professor in the Division of GI, Hepatology and Nutrition at the BCU Health System. Dr. Sanyal, warm welcome. Thank you so much for being here. Please go right ahead.
Thank you, and good morning to everyone. It's a great pleasure for me to talk about a topic that's very close to my heart, which is non-alcoholic fatty liver disease. And today specifically, we'll talk about the proteomic signature of NASH. So here are my disclosures. So a good place to start this conversation is to first define what the disease state that we're talking about actually is. So non-alcoholic fatty liver disease covers a spectrum of disorders, which by definition is not attributable to the consumption of alcohol, thus non-alcoholic. The disease itself is characterized by the accumulation of fat that produces a disease state in the liver, thus non-alcoholic fatty liver disease. Under this larger umbrella term, there are two phenotypes that are defined histologically. One is a non-alcoholic fatty liver, where one gets a pattern of steatosis, where individual cells fill up with fat that get washed out when we stain the slides, and they give this liver tissue a Swiss cheese appearance, as you can see on the left. And it is really not accompanied by much else other than occasionally a little bit of inflammation. On the other hand, there is a more aggressive lesion called non-alcoholic steatohepatitis, or NASH for short. Now, in this condition, you not only have the steatosis, but there is a hallmark lesion, which is hepatocellular ballooning, where individual cells balloon up, as you see those big cells on the right-hand side, and they are often accompanied by accumulation of eosinophilic inclusions around the nucleus, and that's called a Mallory-Denk body. And this is associated with inflammation, and it's typically in a zone 3 distribution. So together, fatty liver and NASH are part of the spectrum of non-alcoholic fatty liver disease. Now, the reason to make this distinction is because the natural course of these disease follow in different patterns. NASH is much more likely to lead to cirrhosis than fatty liver alone. And so once you get to cirrhosis, this is important because it is only when you get to cirrhosis that you start getting liver dysfunction, because the liver has a tremendous amount of reserve. And at the stage of cirrhosis, with progression, one gets liver dysfunction, which is often measured clinically via parts of the MELD score, and by the development of portal hypertension, which can lead to variceal hemorrhage, ascites, encephalopathy, which are the principal liver-related outcomes in this population, and the development of hepatocellular carcinoma, which also can occur in pre-cirrhotic stages of the disease, but albeit at a much lower rate. And then all of these eventually can culminate in mortality. So this journey towards cirrhosis and the development of cirrhosis is extremely important in terms of understanding why this disease is a public health problem. And this journey is characterized by progressive fibrosis. So fibrosis is the hallmark of disease progression. And when we think about the various stages of fibrosis, the stages of fibrosis in progressively capture not only the amount of scar tissue, which in a trichrome stained liver biopsy slide is shown in blue, but also the distribution of the fibrosis, where, as you can see, it is initially in the sinusoids and then progressively involves both the portal areas. You can see the P in the portal area in the second panel, and the V is the vessel, and then there's fibrosis in the middle. Then you form bridges between the central vein and the portal tract, or central vein to central vein, and eventually have complete circumscription of islands of liver tissue or regenerative nodules, and that is cirrhosis. So conceptually, when you think about a normal liver that becomes diseased with fatty liver disease and progresses to cirrhosis, there are two critical things to think about. One is the concept of disease activity, and this refers to all of the injury that is going on from the accumulation of fat that leads to inflammation, and that is basically the driver of the injury and the scarring that occurs. And this is captured under the microscope by the presence of steatohepatitis versus the fatty liver, or through various activity scores, such as the NAFLD activity score, which is a composite of the severity scores for steatosis, ballooning, and inflammation. The consequence of activity is fibrosis and is the disease stage. Disease stage tells us the proximity to cirrhosis and thus to liver-related outcomes and mortality. So in the long term, our goal, of course, is to reduce clinical outcomes and improve survival. But in the short term, our goal is to improve these markers of activity based on the mechanism of action of specific drugs. And then, of course, to also make sure that the disease is not progressing, and that is reflected in improvement in fibrosis or stabilization of fibrosis. And we know from clinical studies that this fibrosis progression has very important prognostic information. And these are data from the NASH Clinical Research Network, which show that the risk of hepatic decompensation events or death from any cause, shown on the right, increase progressively from F0 to 2 to F3, and then from F3 to F4. So you can see really when you get to bridging fibrosis or to cirrhosis, that's when the risk of outcomes really dramatically increase. And this is also together often referred to as advanced fibrosis, or F3 and F4. Now, the other thing we have learned is that as the liver disease progresses, the incidence of non-liver related outcomes also increase. And from this study, we see that the hazard ratio for development of hypertension, new onset hypertension, diabetes, and chronic kidney disease in people with stage 3 and 4 compared to stage 0 to 2 is significantly increased. So if you think about it in more simplistic terms, I always like to think about the journey of the liver in sort of a metro rail sort of format, where if you start with NASH, the progression to cirrhosis, the cirrhosis station is a critical landmark in the natural course of the disease. Now, between NASH and cirrhosis, the causes of death and outcomes are predominantly cardiovascular, cancer, with rare hepatocellular carcinomas. But once you get to that advanced fibrosis cirrhosis stage, now the lines cross and liver-related outcomes become the leading cause of mortality, followed by cardiovascular and cancer-related outcomes. So this switch occurs. And so from a liver perspective, this progression to cirrhosis is really a critical piece and what we focus on in drug development, because that is what prevents those liver-related outcomes. So here we have shown sort of the big picture paradigm of what it is we're trying to accomplish. So we want to capture people with NASH who are particularly at risk of developing cirrhosis and prevent that, and to take people with cirrhosis and to prevent outcomes in those patients. So if we look at the public health burden of disease, these are data from modeling the burden of disease from 2015 to 2030. You can see on the left-hand panel that there are about 1 million people with cirrhosis currently living in the US. And this is expected to rise to almost 3 million by 2030. So the proportion of people living with end-stage liver disease is expected to increase 2 to 3-fold with the aging of the population and continued disease up to 2030. And we know that we only do about 9,000 transplants annually in the US. So this is not a problem that we can transplant our way out of, and thus the need to develop effective strategies. To develop effective strategies, we need to identify who is at greatest risk of developing these outcomes, because everybody is not at the same risk. So we think about this as through this progression from a healthy state to fatty liver to NASH to what we call at-risk NASH, which is presence of NASH, activity score of 4 or higher, and fibrosis stage 2 or higher. And these are the people who have increased mortality, increased liver-related outcomes, and eventually these are the ones who will go on to develop cirrhosis, liver failure, and death. So we want to identify those with at-risk NASH as a very important population for drug intervention, and of course those with cirrhosis as well, although we have not done really great in terms of bending the needle with cirrhosis yet, but it could happen. Now, histology has been the hallmark for learning a lot of this knowledge, and histology has been the conventional reference standard. And the problem, of course, here is that when you want to scale this up as a clinical tool for wide-scale use, one, it is invasive and can be uncomfortable. 10% patients have significant pain, and many patients don't like to have this. It carries risks. Along with the morbidity, it has a 1 in 10,000 mortality rate. And of course, for a diagnostic test, any kind of mortality should be a never, ever event. And in addition to that, the assessment of the liver tissue that comes out is subject to sampling variability, but also observer variability. And the workforce capacity to evaluate, potentially, 100 million people who could be at risk of having fat in the liver to find out who has at-risk NASH is really not an optimal public health solution. So this, of course, is the rationale why we need simple, non-invasive tests that can be deployed at the point of care for evaluation and management of NASH. Okay, so having set the stage as to what disease we're talking about and what is the problem that we're trying to solve, let's next talk a little bit about some basic concepts in biomarker science that will help put all of the proteomic data that we'll eventually talk about in context. So a biomarker is an objective patient characteristic that is measured as an indicator of either a normal or a pathologic process or a biological response to a therapeutic intervention. The functional definition of a diagnostic test basically is one that translates the complex spectrum of disease biology into practical, actionable steps for clinicians to improve patient outcomes. Important to remember, diagnostics make up 4% of healthcare costs, but informs 50% of how the spend is directed. Now, there are many different uses of biomarkers. So when we talk about biomarkers, we must remember that biomarker tools must be fit for purpose based on their intended use, whether it is a diagnostic use, whether it is to assess prognosis, whether it is a predictive biomarker, which tells us if somebody is going to respond or not respond to a certain type of treatment, a disease monitoring context of use, clinical endpoint, or a surrogate endpoint, which may substitute for a clinical endpoint based on biological rationale, prediction of clinical endpoint, and effect of treatment on the clinical endpoint and outcomes. The other piece to think about as we evaluate the utility and overall where a biomarker fits in is biology, the analytical performance, how well does the test detect the biomarker, what conditions are needed for accurate detection, the repeatability, reproducibility, and then its clinical validity. And often many studies will just show you an OROC, which in my view provides information, but really is an incomplete measure of overall biomarker performance. And this, from clinical validity, we go to clinical utility, and then eventually we have to consider benefit versus harm based on the risk of misclassification. We also have to remember that the prevalence of the thing that we're looking for, we're using a biomarker, has a major impact on our ability to exclude its presence or to rule it in. So when the prevalence is extremely low, you will have very high negative predictive values and poor positive predictive values. So just to remind you, and this is Bayesian theory, so just to remind you that even if a test has 90% sensitivity and 90% specificity, which is pretty phenomenal, if the prevalence of the disease you're looking for is 1%, then your best predictive value that you can get, the positive predictive value, will still be in the 15 to 20% range. So depending on the prevalence of and where you are using a given test, that really determines what is the most robust type of information that you can get, and then essentially you work from there. And if you cannot get a high positive predictive value, you may need enrichment strategies. So this is just sort of background information. So there is also a hierarchy of these different contexts of use where you start with diagnostic, and these are used in clinical trials for inclusion-exclusion criteria. Then you move to prognostic, which can be used for risk stratification, leading to treatment response, et cetera, et cetera. So the focus at the very bottom over here is to identify at-risk NASH. This is where we are in the field because we have to identify the pool of patients where we already have now generated the data that they are at risk of great increased risk of outcomes. And those particularly with advanced fibrosis or actually with already have cirrhosis, who even may not have clinical symptoms because you may not get specific clinical symptoms until the disease manifests with a complication of cirrhosis. So now let's move on to the meat of the study, which is the proteomics and the methodology that was used to generate these proteomic profiles. So specifically, of course, we're talking today about the SomaLogic platform. So we know that each protein has a very unique primary and a secondary structure, and the amino acid sequence in these proteins are associated with certain DNA sequences. And so the core concept here is that specific aspects of these proteins and their kind of related DNA are linked through production of aptamers, which bind with high affinity to specific protein targets and are selected for the specific shape from a very large library of such aptamers. And the exquisite specificity allows a scalable platform to be established. So this is already done, this is well established, and this platform has been used in multiple settings. The workflow is as follows. So you have the substrate and you put the reagents on top of it and it binds to the proteins. Non-specific interactions can be dissociated and cannot rebind, and you use a polyanionic comparators to get all this non-specific binding off. So whatever is left on your target is actually a specific protein. And these complexes and dissociated proteins are recaptured, and the remaining reagents are then quantified on a chip. So this then provides us proteomic signatures of the tissue or whatever we are interrogating. Now we move to proteomic signatures of NASH in appropriate context of use, and we're going to speak primarily of the diagnostic context of use first, and then a little bit about disease monitoring and treatment response. So how did we go about this? So what I'm going to discuss with you is a collaborative project that we engaged in with SomaLogic through the NASH Clinical Research Network, which is a NIH-funded research network that I had the opportunity to be part of. And so this is how we approach this. This was published recently in Journal of Hepatology. So everything I'm going to review with you is in the peer-reviewed public domain. So the idea was to develop protein-based current state models in serum with biopsy as the truth standard. So the idea is to do a liquid biopsy and to utilize machine learning to develop binary classification models for each NAS component and fibrosis. Remember, the NAS components are steatosis, inflammation, and ballooning, and then to validate these in specific independent data sets and assess the utility also to monitor longitudinal change during therapy. So while, for example, steatosis, you can go from grade 0, 1, 2, and 3, the big question we ask ourselves is fatty liver disease present? And when we evaluate has fatty liver disease resolved, we really make a dichotomous decision. Is it present or absent? So we drew the line between negative being 0 and positive being anything more than 0, which is 1, 2, or 3. So we're not trying to separate grade 2 versus grade 1 and that kind of stuff here. Similarly, for lobular inflammation, we drew the line between grade 1 and grade 2. Why? Because if you remember, fatty liver is fat with maybe at most 0 or a 1 of inflammation. And the current definition of NASH resolution in treatment trials is that you have to lose your ballooning and you can have any amount of fat. Again, that's why we have fat 0 versus 1, 2, 3, and at most 0 or a 1 on the inflammation. So we drew the line between 1 and 2. Ballooning is the hallmark lesion of NASH. So we drew the line between 0 and 1 so that it gives you a dichotomous readout in the end for decision making. That if ballooning goes to 0 from being present before, then you know you're moving towards NASH resolution as long as the other criteria are met. And similarly, clinically significant fibrosis is stage 2 or higher. So this was how the rationale for doing it this way. So we had a total of about 559 patients, and they were derived from 3 of the NASH CRN cohorts, a natural history cohort, the Pivence clinical trial, which was placebo, vitamin E, and pioglitazone over 96 weeks. And then we had the Flint trial, which was obeticholic acid for NASH. And we applied plasma proteomic machine learned models over there. And so we had an independent model training set. And from this larger pool, we took a subset of 469 patients, samples for validation. And these were, again, 2 different independent validation cohorts. One was a holdout validation cohort that was not included in anything. The other is what we called a paired validation cohort, because remember, Pivence and Flint were clinical trials. So you had baseline and you had end of study samples. So we took half the baseline samples and half the end of study samples and set them aside because we had concomitant histology with them. So that's, for lack of a better term, we call those paired validation data set. And then there was an independent holdout validation data set. Now, the clinical trials also gave us an opportunity to apply these models to longitudinal samples from baseline at different points in time during the trial, at week 16, 32, 48, 64, 80, and 96, with histology at the 2 ends. So with that then, let's see. I lost my pointer over here. Where did my... here we go. Sorry. So this then allowed us to move to the data. So what you see at the top are the data for steatosis in the training, holdout, and the paired validation group. And what you're seeing for steatosis grade 0, 1, 2, and 3 are the predicted probabilities that somebody actually has this. So we set 0.5 as a decision threshold. So anything above that we said was yes. And if it was below, then we said it didn't meet the criteria. And you're seeing the distribution here for steatosis. At the bottom, you're seeing the data for ballooning, which is a little bit You can see the holdout and the paired validation, the data, and particularly for the scores of 2, the data are a little bit cleaner, but there's some overlap over here. Then we move on to inflammation and to fibrosis. And you can see fibrosis, particularly advanced fibrosis and cirrhosis, very nice ability to predict the presence of advanced fibrosis shown in green. Now, this is the summary of the data. So you're seeing the sample size for the training, holdout, and paired validation. You're seeing the number of cases, and then the area under the curve, and the sensitivity, specificity, and overall accuracy of the test. So for steatosis, we started off with, an OROC of about 0.95 and accuracy of 0.86. It did drop off in the validation cohorts, but one thing I'll tell you, in the holdout validation, there were only 8 patients who had no steatosis. So we didn't really have, there was enough patients without steatosis to be able to clearly make those distinctions. Now, if you look at lobular inflammation, it's relatively consistent. It's not unusual from training to validation to have some drop in OROC, and you get from 0.83 down into the 0.7 range. For ballooning, again, you can see 0.87, but in the paired validation, you're getting 0.83, holdout validation in the 0.7 range, and overall accuracy almost in the 0.78 range. Fibrosis looking very good in the 0.85 to 0.9 range. So particularly for fibrosis, a hallmark lesion of progression, really good overall OROCs and accuracy. And then we looked post hoc at risk, because when we started this project, the concept of at-risk NASH didn't really exist. So it was by definition a post hoc analysis where we combined the model outputs for, having steatohepatitis and then having steatosis, inflammation, ballooning, all combined to meet a NS of 4 or higher, and then the fibrosis piece. And that gave us an OROC again in the 0.85 to 0.9 range. So this is very, very encouraging. And if you look at the accuracy again, in the overall accuracy in the 0.7 to 0.82 range. And these are the data for the NAS component predictions and the observed biopsy results. And we're drawing the line over here with the biopsy stage on the x-axis and the predicted probabilities. So you're looking at the fibrosis piece over here, and you can see that on the right, that most of this data are above this predicted probability line. Similarly for steatosis, inflammation, and then also for ballooning, there's a little bit of a tail over here. So all of this actually looks really encouraging, because here's one platform that's giving us a readout on multiple aspects, histologic aspects of the disease. So when we then look at next the model predictions for at-risk NASH versus the histology, you're seeing over here the negative is where the histology is negative, and where the readout is negative versus positive, and you're looking at the composite outcomes over here in the training, the holdout, and the paired validation set. Again, you can see that overall is beginning to separate out. It's not perfect, but it looks really good over here at being able to identify the at-risk NASH population. So one of the key issues in all of this, so all of this is great from a diagnostic point of view. One of the questions that always comes up is, are these things just random associations, or is there real biology that is being captured here? So one caveat is anytime you measure something in blood, it is an integration of what is happening in all the end organs. And so with that caveat, then you come back and look at this, and we look at the principal targets that popped out on the analysis with steatosis. What you see here are the gene symbol, the target name, the UniProt symbol, the T statistic, and the p-value. And you can see these all meet false discovery thresholds. And what you're seeing over here is that there are a number of things from inhibin, prostaglandin reductase, insulin-like peptides, some growth hormone variants, fatty acid binding protein. Several of these are linked to metabolic pathways. The serum IRE1 that is linked a little bit to ER stress, etc. So a number of these actually do show are linked to pathways that are known to be connected to metabolic pathways that are involved in lipid metabolism. Similar, while some actually are novel discoveries as well. Now, if you look at lobular inflammation, again, you are seeing, a number of things such as the immunoglobulin receptors over here, adiponectin, which has been linked also to the inflammatory response. And we didn't see it in steatosis over here as one of the top hits, but it came out in the inflammation piece. Peptide YY, other things over here, serum-associated amyloid. So a number of these are known to be associated with inflammation, chemokines, etc. And then there are a couple that are also new findings. Then you go to ballooning over here. I draw your attention again to a number of these parameters. Of these, the AKR1B10 is very interesting to me. And you can see that the p-value is 2.2 times 10 power minus 16. And this is part of the aldoketoreductase family and really has understudied target. And we published a paper on the transcriptomic profile of NASH. And this came out strongly to be related both to ballooning and disease progression in that paper on NASH transcriptomics. Since then, the Newcastle group published a paper where this again popped out as a marker associated with more aggressive disease. And now we're seeing it as part of the proteomic signature in the blood linked to hepatocellular ballooning. So, there's biology interspersed through all of these, but we're also learning about new pathways and trying to figure out how these all play in. Once again, we published a long time ago that lipoxygenases and prostaglandin biology is important in NASH. And here again, you're seeing a number of signals related to the prostaglandin activities. And then, of course, with fibrosis, many of these are actually linked to matrix and some of these linked also to inflammatory cascades. And there's a growing body of literature on the potential role of the complement in this process. So there's a lot of biology that is also in here and integrated throughout. And these, of course, are going to provide a platform and substrate for specific hypothesis-driven studies moving forward. And we're going to learn a lot from this type of proteomic approaches to really understand the factors that are critically important in disease progression and in development of more aggressive disease phenotypes. Now, talking of disease progression, we've sort of talked about the diagnostic piece. So now let's move on to the disease monitoring and treatment response aspects of this. So first we looked at the Pivence Trial, and this is just to refresh your memory of the Pivence Trial. We looked at vitamin E and pioglitazone and compared it to placebo. Because there were two primary comparisons, the p-value was set at 0.025. And you can see over here, the proportion of people who improved steatosis is significantly greater than placebo for both vitamin E and pioglitazone. Similarly, and these are the p-values over here. Similarly, when you look at the improvement in ballooning, you see the proportion of people who had improved ballooning significantly greater in both vitamin E and in pioglitazone compared to placebo. And when you look at fibrosis, there's a higher proportion of people numerically in both pioglitazone and vitamin E compared to placebo, but these did not reach statistical significance. And part of it is this study was not really powered to detect this effect size. So for this effect size, if we had a 3 times larger sample size, this would have reached a statistical power. So if you think of the size of many of the current phase 3 trials that are going on, as opposed to the Pivence trial, with this effect size, you would have actually started seeing some statistical significance as well. So keeping this at the back of your mind, now we move on to look at the proteomic signatures that we potentially saw in these patients. So based on our models, where we again set the threshold at 0.5, we're reading a probability score that the model has changed from, before to at the different points at which we measured these tests, that they have fallen below those thresholds for steatosis. And you're seeing this response in steatosis, in pioglitazone, shown in the dark blue, vitamin E, little bit less, and the placebo is relatively flat, also indicating that the proteomic signatures are relatively specific. So they were able to cleanly separate out the placebo arm from these treatment arms. Similarly, when you look at ballooning on the right, the placebo remains relatively flat, and you're seeing a very nice reduction in the ballooning shown on the right. We see an improvement in inflammation, and you're even capturing the small differences that we saw histologically in fibrosis. And remember that these signatures, the proteomics, a priori independently predicted this without really, it was not modeled. The histologic outcome was not part of the model. That was the validation piece. So the model predicted whether fibrosis is getting better, model predicted if inflammation is getting better, model predicted if steatosis is getting better, and then we checked to see whether indeed it got better or not. And so this is what you see. That's why I showed you the histology data just up front, just to set the stage and as a frame setting concept. So now if you move on to the Flint study, what we see over here are the histological outcomes. So you're seeing the primary endpoint, which was a NAS improvement of 2 points or more, and the proportion of people who met it. Blue is OCA. The open bar is placebo. You're seeing improvement in steatosis, lobular inflammation, ballooning, fibrosis, and then NASH resolution, and then portal inflammation over here. So here you can see the changes in steatosis are more modest. And remember, though, although histologically we did see improvement in steatosis in the Regenerate trial, this did not necessarily translate into a big difference. The data are what they are, but you do see a modest improvement. With ballooning over here, looks like the OCA group had a little more ballooning to start with, but they did show this improvement in ballooning compared to the placebo, which was relatively flat. Similarly, you can see a nice improvement in inflammation. And then with fibrosis also, you can see at beginning, the fibrosis scores were higher and they trended down over time, so that at week 72, when the biopsy was done, the fibrosis, was lower compared to the placebo arm. So there is some overlap over here, but basically the trend lines all look they're moving in the right direction in terms of the ability of the proteomic model to predict the histologic response. Now, what we've shown you here is that first, that the placebo response arms remain relatively flat. So in a blinded manner, or masked manner, the model was able to identify that these are the placebo arm of patients, and they didn't really change that much. And were relatively flat and tracked the histology. The second is across 3 different mechanisms of action. So you have pioglitazone, you have vitamin E, and then you have OCA, that in general, these are tracking the histologic response in these patients at an individual patient level concordant way, in a patient level concordant way, which is extremely encouraging. And I think this is really the first time we have longitudinal data using a blood-based biomarker that is actually tracking the outcomes with the key histologic features of NASH in the setting of a well-constructed, rigorously done set of clinical, not just one, but a set of clinical trials. Now, talking of clinical trials, those were all studies that were done through the NASH CRN, which is a network of 8 academic centers with a very deep interest in NASH, and a pathology committee, which has 8 members, and they read histology in a very specific way. with a very defined protocol. So maybe in a other setting, those data might not be quite as translatable. So these are data from a recently published or presented Falcon trial. So this is a post hoc analysis from the Falcon trial. So to remind you again, the Falcon trial looked at FGF21, pegmelpharmin, and this is in a patients with bridging fibrosis, and Falcon 2 was in cirrhosis. And essentially, the idea here in this post hoc analysis was to see whether all the different biomarkers and the histology, what was the level of concordance between these? And so what you see over here at the top is the primary histologic endpoint, which was fibrosis regression. Then you have the Ishak fibrosis stage, which is modified for NASH. Then you have NAS lobular inflammation, ballooning, NAS overall, liver stiffness with MRE, ELF score, ProC3, PC3X, LDL. Then you have the soma signals, and then a whole bunch. You can see AST, ALT, NAS, steatosis, FIB4, etc. So group 1 are people who are these are people who are non-responders. So non-responders are in black, responders are in yellow. So if you look all the way to the right now, here are people who predominantly are responders, where everything got better. And you can see over here that the soma signals actually picking these out. And on the very left side, picking the non-responders out very well. So I have arrows to point out at the proteomic model, which is the NASH steatosis. You have the inflammation, you have fibrosis, you have ballooning. And so over here, if you look on the left, you are looking at all this dark blue and black. So it's picking this up very nicely. At the top over here on the left is the histology that matches this. And if you go to the right, if you look at people who are complete histologic responders over here, you can see the soma signals picking it up. There is a population in the middle which is noisy, and there needs to be more refinement here. But again, it shows that the overall performance is actually very, very encouraging. And I think this is actually an important step forward in the field to try and better define non-invasive methods of evaluating histologic response. So to wrap up, in summary, proteomic analysis of serum can identify a signature for key features of NASH, that is steatosis, lobular inflammation, hepatocellular ballooning, and fibrosis. These are related to biological pathways linked to NASH, and also several novel targets and pathways were identified. In independent validation cohorts, the signatures could be reproduced. These measures were also sensitive to change and recapitulated many of the histologic changes seen in placebo and treatment arms of several clinical trials. These proteomic signatures carry the potential to serve as important diagnostic and disease monitoring / treatment response assessment tools. While much work still remains to be done, these are very important steps in the field and foundational for the future work that remains to be done. So with that, I will conclude, and thank you very much for listening to me, and I look forward to your questions.
Thank you so much, Dr. Sanyal, for this excellent presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for today. So let's get started. It looks like we have a question about ProC3. Was this detected anywhere in the data set?
No, we did not measure ProC3 in this data set, to my knowledge. But maybe somebody from SomaLogic can answer. But to my knowledge, it was not on the list of the proteins that were measured.
Yeah, I don't know off the top of my head either, but we can certainly delve into that a little bit more. If it's not on the platform, we can share some more information about what we may know about anything related. Do you have any thoughts, Dr. Sanyal, about what was seen in the proteomic signals sort of on a pathway level with ProC3 or anything that might indicate that there was some association?
So I want to remind people that ProC3 is a collagen fragment. So when you are actually having fibrogenesis, you have, during both fibrogenesis and fibrolysis, you have while the collagen is being made and broken down, small pieces of collagen break off and can go into circulation. And ProC3 specifically was developed as something which is a marker of the fibrogenesis activity. And so it is not a biological pathway itself, it's a fragment indicative of fibrogenic drive, if you will. And so in this particular study, if I can go over here and bring this back up over here, some of the things that we saw were linked to these are all proteins that are linked to connecting inflammation and matrix. You have colectin, you have neurophasin, you have complement, FC receptor-like protein, VEGF, which has been linked to fibrogenic drive. There's a procollagen dioxygenase 3, which is probably the closest one to the collagen synthetic pathway itself. Then there are protein signatures linked to Wnt that were in here. So these are the ones that actually popped out over here in this analysis. We do see procollagen endopeptidase enhancer in the lobular inflammation piece. And the Adams-like protein 2 is one of the few that are common between ballooning and fibrosis. So I think that's another one that is worthy of keeping an eye on, because this may be another biomarker that links the activity to the fibrosis readout.
Yeah, terrific. Thank you. Related to that, there's a question specifically about macrophage markers. Were any of these detected?
So there were a number of biomarkers detected, but in terms of using them as a diagnostic signature, they did not make it into the cut at the very top. These p-values for false discovery, etc. So, as you go down this list, the p-values and the false discovery rates tend to change. So we sort of initially set this relatively, conservatively, but the full list is actually provided with the manuscripts, and one can look at that. And there were things in there, but from a diagnostic signature point of view, they did not, rise to the top of the list to support its use as a diagnostic, panel.
Yeah,
does that make sense?
Yeah, definitely. Thank you very much. It's important to make those distinctions.
Yeah, I think it's important to remember that the biology, the inflammatory process is very complex and noisy with a lot of different pathways and signals involved. So what we were trying to do over here, which is a biomarker exercise for diagnostics, is to take the signals that rose above the noise with enough fidelity to warrant trying to create a panel around.
Terrific. Great. Thank you. There is a comment. Great presentation. Thanks very much. What is the most translatable preclinical animal model and the most reliable biomarkers of such models of NASH? And there's a follow-up too, but we'll start there.
It's a loaded question. So, preclinical models are only as good as the question that you are trying to ask. So if you want to learn a specific pathway, there are various overexpression or loss of function models that are available. There are also now a number of iPSC-derived models. There are small in vitro models where you take hepatocytes, macrophages, macrophages and stellate cells, and put them into chambers with flow like the HemoShear model. There are other organoid models that have been developed. Then there are mouse models, and probably the ones that are closest are the diet-induced obesity models, which replicate the systemic milieu in which the fatty liver disease develops. The models where the liver, I can tell you which models are not very good, in my opinion. Models like the MCD model, where you can get a very nice liver phenotype, but the overall systemic milieu where the animals don't gain weight, you don't develop the systemic inflammatory and insulin resistant state and obesity and adiposity, that probably is not a great model. Models that are dependent on knocking a specific gene in or out, models that are dependent on giving poisons like carbon tetrachloride to induce fibrosis that no human being will ever see, are less translatable. The closer the model comes to reflecting the conditions in which the human disease develops, and there have been a number of recent publications sort of looking at all these different parameters. So I would recommend that you look up. There's a paper in the last couple of years looking at. I'm blocking on the first author, but it was in hepatology, I believe, looking at all the available models and sort of comparing them. And so in general, the diet-induced obesity type models came out on top. It's important to also remember that there are diet-induced models which use very high levels of cholesterol, and that are, again, creates another layer of difficulty in translation to humans, although in general they reflect sort of the same disease phenotype. So that's the preclinical model question. There was a second follow-up question, wasn't there?
Yeah,
sort of that.
No, that was terrific. The second part of the question was, is there any scope for new biomarker identification in animal models to accurately predict the disease progression in addition to the histopathology findings?
Oh, absolutely.
So
The short answer is yes. And the long answer, of course, can be very complicated. But I think if you pick models that that meet the systemic milieu, the insulin resistance related parameters, who develop the histology and the fibrosis progression to advanced fibrosis or early cirrhosis, and those that have a transcriptomic profile that is strongly concordant with human disease at each stage, because even within the same patient, the transcriptome changes as the disease moves from one stage to the next. If you get that type of model, those provide very powerful opportunities for additional, novel biomarker discovery. By no means have we solved the biomarker issue yet. I think there's lots of room for more work and novel biomarkers.
Yeah, excellent. Thank you. Just one more specifically related to preclinical models is fructose-induced NASH in zebrafish as a model to test steatohepatitis. How robust is that as a model?
It's a high throughput system to see whether you get steatosis. And the key, and I have not seen that data, and that's just me, that doesn't mean it doesn't exist, is really a strong concordance between the zebrafish models and the human transcriptome and fibrosis progression signals. Getting fat to accumulate is pretty easy, across a variety of mechanisms. Showing actually that it leads to injury and fibrosis and that it matches the human disease progression, those are critical elements in terms of maximizing the likelihood of translatability. Even with all these caveats, there's always a gap between preclinical models and human. Our approach, therefore, has always been, instead of moving from preclinical to human, is to actually start with the human, define the disease state, and then try to find the preclinical models to get them as close to the human, and then to do back translation to understand how specific pathways and other things behave in the preclinical models, because you can do things in those models that you cannot, for obvious reasons, do in humans.
Yeah, that makes great sense. Thank you. So moving now to a little bit about the signatures, would these signatures be relevant for multiple different mechanisms targeting NASH to measure treatment response?
So we have got data. I can only respond to what we have data on. So we've got data on vitamin E, pioglitazone, and OCA, and these tend to track in these populations. Whether they will apply to other mechanisms of action, we'll need to generate the data to show. Typically, if you look at how biomarkers were developed, for example, hemoglobin A1C for diabetes, once you have data across multiple mechanisms of action, widely disparate mechanisms of action, I think then you're getting closer to having something that meets the biological aspects of is one of the checkboxes for on the pathway to developing a surrogate endpoint. But I think this is very encouraging because, to my knowledge, we have not had a biomarker panel to date that across multiple mechanisms of action can separate the placebo responses from the drug responses.
Yeah, it is very exciting. Along those lines, can you comment a little bit about the populations used in these models and the prevalence in different ethnicities of NASH and how generalizable these models may be?
I don't think we have the data to make large population. This was 560 patients from the NASH CRN. So the NASH CRN is a NIH-funded consortium in academic centers with all the ascertainment bias that goes on in those kind of centers. It is predominantly a Caucasian cohort, very small number of Hispanics, and even smaller number of African Americans and Asians. So there is still an unmet need to test the robustness of the diagnostic utility of these and the disease monitoring utility in larger cohorts. I believe in litmus, this is being looked at, but again, I remind you that litmus is a Western European consortium. So again, you're getting more data with the predominantly Caucasian cohort. So there is certainly room to look at larger cohorts and in greater populations, particularly Hispanics, because they are at risk of more aggressive disease. They do tend to they have the underlying PNPLA3 mutation. They have other things going on that really put them at risk of getting more diabetes, more liver disease, more outcomes. And so that's a huge unmet need. I think that's still present. And African Americans, even though the prevalence is very low, but there are people, it's a common misconception that African Americans cannot get the disease. Their risk is low, but we have seen African Americans also who have significant NASH, and how these models perform on those in large enough numbers of African Americans also remains a little bit of an open question.
Yeah, thank you. So important to pursue that work. How do the individual component scores correlate to comparable clinical assessments like inflammation score versus ALT or steatosis score versus fat fraction on MRI PDFF?
So we don't have a lot of PDFF data. Remember, a lot of these MRI tools were developed after Pivence, and these are all based on sample sets that are a little, that were sort of predated before we got going with a lot of MR-based measures, et cetera. So cannot answer that question. Even the fibroscans came much later into the story compared to when we started with this sample sets. This has been a long journey, by the way. And just to remind everybody, the initial, all of this really started actually quite a few years ago. So this has been a long time coming. So everybody thinks you can just go get samples and off you go, and in 3 months have the results. This has been quite a process to get to this point. We don't have that data, unfortunately, and again, would love to obviously, that's a very relevant question, and we need to look at that. Now, ALT is very interesting. So what we are measuring here are the aptamers, the protein itself, whereas the assay that we measure clinically is a functional assay where you take the serum and you have a substrate for ALT and you get a color signal, and the rate of color development determines is read off a spectrophotometer, and and there is a standard curve from which the AST/ALT values are estimated. So we did not actually see a correlation with AST/ALT, but the measures are completely, the way they measure, it's a little bit apples and oranges, because what you're measuring here is the presence of the protein and not necessarily its functionality. So it doesn't really exactly correlate, but we did not see, and it's in the manuscript as well, that we did not see this correlation with AST and ALT.
Yeah, that makes sense. So we have time for one more question. We really would like to know what is next for your research and proteomics applications in these areas of high clinical unmet need, and what do you think is needed to help establish these models as screening tools for clinical research and diagnostics?
I think we need a larger and a more diverse diagnostics to test these in a larger and a more diverse population for diagnostic purposes. We need more data on prognostic, and then hopefully some of the clinical trials that are including these panels for evaluating treatment response will give us additional data on disease monitoring. Together, I think these will help move the proteomics platform along, to answer the key unmet needs within the biomarker space. For my own personal research, we're doing a lot of work with a number of biomarkers. So we also have ongoing studies with fibrosis biomarkers, such as the ADAPT score, which is modified from ProC3, with NIS4 and now NIS2, and then with ELF score. So we have a very deep interest across a broad range of biomarkers, and we are particularly, now that we have imaging tools as well through the NIMBLE consortium, we hope that in stage 2 of NIMBLE, we will be able to look simultaneously with ultrasound-based tools, MR-based tools, and circulating biomarkers to address some of these key unmet need contexts of use.
Yeah, excellent. We really look forward to that. Thank you so much again for your time today, Dr. Sanyal, and your really critical and exciting research. We'd also like to thank LabRoots and the sponsor SomaLogic for underwriting today's educational webcast. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, have a great day.